on next Tuesday, November the 6th. Make sure you vote before you come to your test. I want to make sure you guys vote.
I voted.
Good. I already did too on Saturday, okay? I have an update for you all. Okay. I know some of you have come to me about trying to access Mastering Microbiology. Some of you even bought a code. Those codes are really expensive. And I said, get a refund. I said, don't spend $50 or plus on an access code. That is absolutely ridiculous. I have codes for you, but it took a while for me to fight for them. In other words, it's easy to get free codes if the class is online, for obvious reasons. If the class is in person, it's very difficult to say why you need codes for an online service. So I had to really go to bat for you to get free codes, whether you bought the new book or not. Okay? So I will give you the codes via email. That way you get onto Master Microbiology and you can play with it. Okay, I had to say basically I will give you assignments. I will not give you assignments, but I had to say that to get codes for you.
Oh.
You're welcome.
Yes,
so it's free. Use it. The codes, they do expire November 15th. So as long as you use it before the 15th, then you have access until the end of this course, which is December. But if you wait until after the 15th of November, your code will expire, and then I can't get you another one. So I will send you a code today via Angel. It's for free, so you may use it. Thank you.
Thank you.
You're welcome. So just do the assignments, but they won't count for anything. Real quick, we were talking about virulence factors, okay? In other words, we're talking about pathogenicity and then virulence factors. They go hand in hand. If an organism is said to be pathogenic, it just means it's able to cause disease in a particular host. Okay? Now, in order to cause disease, okay, that organism has to be said virulent or have virulence factors. In other words, in order to cause disease, you need virulence factors. In other words, these factors allow the microorganism to what? Attach to the host, right? You have to attach. You have to get into the host. If you're into the host, you have to what? Evade the immune system, correct? Not only that, you need to do what? Obtain nutrients from the host. All of these virulence factors deals with all of that stuff. In other words, get into the host, right? Evade the immune system and get nutrients. So I mentioned these, I think, on Thursday, correct? So let's get into them one by one. Okay? Now this chart is simply going over organisms based on their virulence. In other words, how likely are they going to cause disease if they come in contact with you? Now, the less virulent guys, like lactobacilli, for example, or Candida albicans, this guy is really not pathogenic at all. But this guy here, Candida albicans, that is said to be what? An opportunistic pathogen. It's less virulent, meaning you have to be either immunocompromised or something else in you is off balance in order for that yeast to cause a yeast infection. Most of you have it in your mouth or on your skin. And as long as you're healthy, Candida will not cause infection. So if something's off balance, for example, it can cause thrush, right? That's yeast infection in the mouth or yeast infection in the vagina. Now, if you go on down off the line here, you're saying it's pestis, right? That's absolutely very virulent. If you get that bug, you got basically the Black Death, or basically bubonic plague. This one here causes rabbit fever. Now, here's the deal. Just because this particular organism is the most virulent, meaning if you get exposed to it, you'll get rabbit fever. It comes from rodents. That's why it's called rabbit fever. It comes from rodents. That disease is not particularly, it's not bad. In other words, you can get rid of it fairly easily. So the take-home message from this slide is this. Even though an organism is said to be very virulent, meaning if you're exposed to it, you will get the sickness, it doesn't mean that that sickness is absolutely severe. Is that okay so far? Okay. All right. Real quick. Extracellular enzymes. These are what? Virulence factors. These are factors that a pathogen needs to have in order to make you sick. Now these extracellular enzymes, they are made within the cell, correct? And they are secreted on the outside and they cause harm to the host on the outside of the bacterial host. Typically, they dissolve some structure or some chemical in the body, and these factors help either to maintain infection, invade deeper tissues, or avoid the immune system. So let's get into those really quickly. All right. Now, most of these will be in picture form, because as you know, I like pictures. But one thing you need to know is highlight hyaluronidase. This guy here, you need to know how this works. So hyaluronidase, that is an enzyme secreted by some pathogens. And it allows some pathogens to basically dissolve the, quote, glue that keeps cells together. So normally, for example, in your tissue, if these are epithelial cells, these cells are stuck together. There's some glue holding the cells together. Well, if this bacterium secretes hyaluronidase, which is an enzyme, is going to dissolve away the glue. What does that mean? If you dissolve away the glue, that means these cells are no longer attached to each other. And allow these bacterial cells to what? Go deeper into your tissues. So far, so good? They are invading deeper into your tissues, either in your skin or within the mucous membrane. It's called hyaluronidase. Another set of enzymes you should be aware of is coagulase and kinase, especially coagulase, because you will see that with Staph aureus later on in your laboratory. What does coagulase do? Probably forms what? Clots. Now, why would, for example, Staph aureus secrete coagulase in your blood system to form a clot? What is it trying to do? It's trying to hide, right? It is trying to hide from your immune system. So if you look at this blood clot here, those bacteria cells are within that clot. Your immune system can't find them. It's like an artificial capsule, correct? They're hiding. That's how they evade the immune system. Now, let's say they want to get out of the clot. They don't want to stay in the clot, right? What if they want to get out of the clot to invade deeper tissue? Well, they need another enzyme called a kinase. So the kinase is the opposite of coagulase. If coagulase causes blood to what? Coagulate, meaning to form a clot, then kinase will be the opposite of that. Kinase dissolves the clot away. It allows, for example, Staph aureus to get out of that clot and go somewhere else to obtain nutrients. So those enzymes, you should know how they work. These are extracellular enzymes. These are what? Virulence factors. Here's the deal. You have to understand that non-virulent bacteria, they wouldn't have these things, right? If you're non-virulent, you wouldn't have these enzymes. You wouldn't need them. Only the pathogenic guys, they need them to be able to cause infection and cause disease. Now let's talk about toxins. Now, just like those extracellular enzymes for the most part, most enzymes are proteins. These toxins are also protein-based substances. Okay? So keep that in mind. Now, what do these toxins do? They either directly harm the tissue, we'll talk about that, they either directly harm the tissue, they directly damage the tissue, or they trigger some immune response. And depending on what type of toxin you're talking about, it's either going to harm your tissue or cause you to have a fever, for example, or cause inflammation. Now, toxemia just refers to toxins that are released in the bloodstream, and those toxins are carried all throughout your body so that you feel sick all over, makes sense? So instead of you just having, for example, a swelling on your arm, if the toxins released into your bloodstream, you end up being sick all over. That's why you feel fever, for example, or malaise. Now, we'll talk about two types of toxins. Exotoxin meaning out, correct? And endotoxin probably meaning within. All right. Now, how do exotoxins work? Exotoxins, just like those extracellular enzymes, they are made within the cell and they are secreted outside of the cell. That's why they're called what? Exotoxin. Now, both gram-negative and gram-positive bacteria, they make exotoxins. I repeat, both gram-negatives and gram-positives, they will make exotoxins. Now, in terms of how they work, it just depends on the type of toxin. For example, this figure is showing you a cytotoxin. Now, cyto means what? That prefix, cyto means what?
Fluid.
Means cell. Whenever you hear cyto, that means cell. Lysis, but not lysis, excuse me, in this case, toxin is a toxin. So a cytotoxin is what? Killing cells. It's directly killing the cell, like this guy over here. These cells are dying, which allows these bacteria to invade the tissue. That's just one type of toxin. There are two more you need to be aware of, and we'll get to that when we get to that table. So be aware of a cytotoxin. It kills the cell directly. Also, these guys, since they're excreted out of the bacterial cells, they produce an immune response. So with these exotoxins, since they are secreted outside of the bacterial cell, they actually produce an immune response. In other words, you produce antibodies against these exotoxins. So far, so good? And since you do produce antibodies against these exotoxins, that means we can actually make vaccines with deactivated toxin to do what? Stimulate an immune response. This is how this works, correct? Now, what about endotoxins? Here's the difference now. Only gram-negative bacteria, only gram-negatives have endotoxin. We talked about this in some other chapter. In other words, the endotoxin is simply the lipid A portion in their LPS, the lipopolysaccharide. Where or when do we mention LPS? Where is it located in gram-negatives? Where, Justin?
The structure outside.
Outside where?
So on the outer membrane?
It's in their outer membrane. So in other words, it's within the cell wall. It's within the outer membrane of gram-negatives only. That's why it's called an endotoxin. They don't secrete it out. It's in their outer membrane. It's in their cell wall. So it's in them. They only release it, A, if they're divided, for example, it may get released. B, if they're dying, they'll release that lipid A portion of the LPS. Or C, if they're being phagocytized. In other words, if a white blood cell is actively eating them up, well, inevitably, that lipid A of LPS, that's going to be secreted, and that's the endotoxin. In other words, it's not secreted. It's a part of the gram-negative cell membrane and cell wall. It's only released when the cell is either dividing or it's dying. It's a lipid. So it does not trigger an immune response, okay? So you do not make antibodies against lipid A. And because of that, we cannot make vaccines against lipid A. It's a lipid. So far, so good. Proteins, we can. Lipids, we cannot. Also, lipid A, for example, it causes generally fever, inflammation, muscle fatigue, malaise, all the above. In other words, it's a generalized symptom. Not like with the toxins. I mentioned toxins are very specific in terms of what they target. Lipid A causes the same, whether it's from E. coli or from salmonella. Fever, inflammation, overall just feeling sick. In this table, we're comparing exotoxins with endotoxins. I've highlighted the things I've talked about in terms of where they're found, how they're made or where they're located, their chemical nature, their toxicity. In other words, these have a high toxicity because they actually do some damage to your tissues. The lipid A portion of endotoxins, they have a low toxicity, but they just kind of make you feel overall bad. All right. The effect on the host. I've highlighted the other types of toxins, and I do want you to know how they work. I talked about cytotoxins, they kill cells. A neurotoxin. Neurotoxins are made by bacteria like Clostridium tetani, for example, or Clostridium botulinum. Okay? They make neurotoxins. If you're talking about tetanus, or basically lockjaw, right? That toxin prevents it keeps your muscles in a contracted state. That's why it's called lockjaw. Okay? Likewise, botulism, that's the opposite. Okay? And botulism is a form of paralysis. Those neurotoxins prevent your muscles from contracting. In other words, you can't breathe, makes sense? If you can't breathe, guess what? You go die. All right? So both of those guys, those are examples of what? Neurotoxins. They affect the nerve cells directly. How about an enterotoxin? What do you think an enterotoxin is targeting? Entero. Think of enteric bacteria.
Intestines.
In the intestines, right? In the gut. So think of all those foodborne pathogens like salmonella, Staph aureus, E. coli, for example. Not E. coli, but salmonella and Staph aureus, for example. They can make enterotoxins. These attack what? The intestinal tract. So think of your foodborne illnesses, got it? Think of diarrhea, stomach cramps, think of that. They attack that system. Fever producing, I mentioned what? Endotoxins? Yes. Okay, that's primarily what they do. They cause a fever. And antigenicity, that just means can they produce antibodies? Can your body make antibodies against them? since the exotoxins, they are what, protein-based, okay? Your body can make antibodies against it, and we can make vaccines against that. With the endotoxins, it's lipid-based, okay? Very weak response. We cannot make vaccines against lipid A. What do you do? I'm not going to hold you to types of diseases, but I've already mentioned botulism and tetanus, for example. Those are neurotoxins. Cholera, for example, that's a type of enterotoxin. Diphtheria, that would be an example of a cytotoxin, so actually killing the cells. How about avoiding phagocytosis? In other words, avoid being eaten up by the white blood cells. Let's see how we can do that. Okay. Now, we've already talked about capsules long time ago, correct? Probably chapter 3. Okay. That capsule has two ways of avoiding phagocytosis. One, that capsule is basically sugar, right? It's basically polysaccharide. The same polysaccharide you can find in your body, which means your immune system sees that polysaccharide and it doesn't recognize it as an invader, makes sense? The sugars on that capsule is basically the same sugars you can find in your body. So that's how the bacteria basically evade the immune system, if they have a capsule. Likewise, that capsule is slippery. It's basically a sugar coat. Think of it like soap. If your hands are wet, correct, sometimes you have a difficult time getting soap if it's wet. These capsules are so-called wet. So phagocytes, if they're using their pseudopods, correct, they're literally trying to do that, right? They have to grab onto the bacteria. So good, they have to do that. They have to grab onto them. Well, if the capsule is, quote, wet or slippery, those phagocytes just have a hard time getting their hands around that capsule. So the bacteria can actually evade phagocytosis that way. So far, so good? Now, what if the bacterium actually got phagocytized? In other words, it got engulfed by that white blood cell. What are the options now? Well, let's see. Some bacteria actually produce proteins that will prevent a lysosome from fusing with that vesicle that was formed from phagocytosis. What you have to understand is this. When you do phagocytosis, you have now made a vesicle, correct? That's a vesicle. That vesicle has no enzymes, right? So you need something with enzymes to digest the bacteria. So far, so good? Now, the vesicle that has enzymes, that's your lysosome. Think of Lysol, right? Think of trying to break something down. The lysosome actually has enzymes. So far, so good? Okay. Now, that lysosome will actually come and fuse with that vesicle that was formed as a result of what? Doing this. And that's how you actually break down the bacteria. Well, some bacteria, even though they were phagocytized, correct, they prevent the lysosome from fusing with that vesicle. So they actually end up growing within that vesicle they were taken in. They actually grow inside. They never die. That's one way they can do it. Some bacteria, they literally make a leukocidin that actually kills the white blood cell. Now, how do you know a leukocidin kills white blood cells? It's all in the name, makes sense? Leuko is referring to what? White blood cells. Cidal, right? Cidal means what? To kill, right? So a leukocidin, that's actually killing white blood cells directly. Here are some pictures. If you couldn't follow what I was trying to tell you about, this is basically, this guy is so slippery, those pseudopodia cannot grab onto it, okay? So this bacterium with the capsule, it gets away. But let's focus on this. This is said to be incomplete phagocytosis. In other words, this phagocyte ended up doing that, right? It engulfed the bacteria. So far, so good? Say yes or no.
Yes.
Yes. Okay, y'all gotta talk to me. I can't read your mind. So there's the vesicle, right? And there's your bacterium. But in order for him to die, right? I said this vesicle does not have enzymes. You just did that, right? There are no enzymes in there. This lysosome actually has the enzymes to digest that poor bacterium. And usually the lysosome would fuse with that vesicle to release out the contents, correct? Some bacteria prevent that. If they prevent this fusion, they get to multiply. They get to reproduce. Hit it.
Where are the materials coming from for that bacteria to reproduce? Because if they're preventing fusion, then aren't they just kind of trapped in there with very, I mean, like, wouldn't it eventually use up all the resources, or are things entering that vacuole?
Things can enter that vacuole because any vacuole, it has a membrane. Just like this out here is a membrane. Things can go in and out. Just like if this was the actual cell, things can go in and out. So things can go in and out of that vacuole just by the process of diffusion.
So it's not truly isolated.
It's not truly isolated.
Right.
It's just a membrane. Things can go in and out.
Okay.
So they can get food, and they're just going to multiply in there. Think about some of the bacteria I mentioned, and think in chapter, I think it was 12. Some bacteria actually survive in white blood cells. This is what they're doing. They're just chilling right there, having a good old time. They're not dying because they are preventing this lysosome from fusing with that vesicle so that they can just be dissolved. Any question about that process in terms of how they avoid being eaten up, basically, or digested once they're inside of the phagocyte? Okay. Now let's talk about the actual stages of disease. Now again, I mentioned infection last week. Infection just means you have the pathogen inside of you, and it can be growing, but it has not made you sick. That's not the same as disease. I don't want you to get this wrong on the test, right? Infection means you have the bacteria, or the fungus, or the worm, it's in you, and it's growing.
Like typhoid Mary?
Right, like typhoid Mary. She was infected with salmonella, exactly. But she never got the disease, makes sense? You only get the disease when your immune system gets caught off guard, makes sense?
It's like the distinction between TB carrier and TB disease.
Right, you can be a TB carrier, you're infected with tuberculosis, right? But you don't have the disease. Okay, very good. All right, so let's talk about the actual stages of disease, okay? Disease, which that's the real term, right? Disease follows what? Infection. Okay, you only get disease when your immune system is what? Caught off guard, your immune system is compromised, and that pathogen can basically take control. What are the stages, okay? First is your incubation period, and of course I'll tell you more details on the next slide. Okay, first is incubation period. Think about what's going on there. Next is the prodromal period, illness, decline, and convalescence. So on the next slide, I'll tell you what's going on in those 5 different stages of disease. Disease follows infection. First stage is what? The incubation period. What do you think is going on in the incubation period? Okay, so the bacteria are actively multiplying, okay, but they have not reached high numbers to trigger an immune response. They're just growing. They're just multiplying. Now, in terms of how fast they multiply, and typically the incubation period, it is between what? Infection and the first sign of symptoms. That's your incubation period, makes sense? So incubation is basically between infection and your first sign of symptom. That's incubation period. Now that depends on the type of bacterium in terms of its virulence. Some bacteria are more virulent than others. If they're more virulent, they get to evade the immune system and they get to multiply. How about generation time? In other words, how long does it take a particular bacterium to go from 1 cell to 2, 2 to 4, makes sense? If they have a high generation time, like E. coli, for example, okay, they're going to have what? A short incubation period. That's why most foodborne illnesses, right, you get sick pretty quickly. If it's E. coli or Staph aureus, they have a short generation time, which means once they get inside of you, they can grow very quickly, and they can get to high numbers to the point of what? Actually making you sick. Also, the incubation period depends on the host immune response. In other words, are you healthy, right? Or are you immunocompromised? If you're healthy, you can probably kill those bacteria immediately, correct? However, if you're immunocompromised, okay, you may have a very short incubation period because your immune system is doing nothing at all to combat those bacteria that are what? Actively growing in you. That's incubation period. Now, if the organism gets past that, and note your time. This is time, right? This is what? Number of microorganisms or the intensity of your symptoms. And notice here, they're kind of slow right here, right? They're kind of growing. Here is the prodromal period. This is basically the first sign of symptoms. Usually, you feel kind of tired, right? Your muscles kind of ache. But the symptoms are not bad enough for you to miss class. Makes sense? You kind of feel like you're getting something, but you come to class anyway. That's the prodromal period. Not all diseases have this prodromal period. Some go from incubation right up to illness. means flat out all your symptoms. But if you have a prodromal period, that's your first sign of symptoms, very weak or vague, maybe tired or muscle aches. Okay? After that, you go on to your illness. Know what's going on here, right? Lots of bacteria, right? Lots of symptoms. Illness is basically where your symptoms are the most severe. This is where you probably go see a doctor at this point. In other words, you made it through prodromal, right? You were okay, but now you're feeling really bad. You got a high fever, correct? Throwing up possibly, something's not right, going off a couple of days. This is the point you probably go to a doctor, you probably get some antibiotic, make sense? Okay, so this is your illness. But look what's happening, right? Lots of bacteria there, right? That's why you feel so sick. Not only that, at this point here, your immune system is still trying to catch up. Okay? So far so good? Your immune system is still trying to catch up to the pathogen. That's why their numbers are so doggone high. That's why you feel bad. Now usually you get some antibiotics to give your immune system a boost, correct? And then you go to your decline phase. At this point, this is where either your immune system is now taking control, right? Either A, your immune system is now attacking that cold virus, because again, if this is a viral infection, there is no antibiotic that can save you. Your immune system has to deal with it. So if you're talking about the cold or the flu, this point here is where your immune system is now taking care of that virus. If you're talking about a microbial or bacterial infection, this is where, for example, you've gone to the doctor, right? And now you're taking what? Your antibiotics to give your immune system a boost, correct? This is your decline phase. Your signs and symptoms, they start to go down. After that, you enter convalescence. And convalescence, this is basically recovery. Your body is now healing from the infection. Note the number of microorganisms, right? Pretty low, right? This is the healing process. Now, in terms of how long this healing process is, it depends on your illness, right? If it's something like the cold, not long to recover from a cold, right? But if it's something like leprosy, which actually forms lesions on your skin, for example, it takes a long time for your skin to heal. So that convalescence depends on what did you have in the first place. But here's the deal. No matter what stage you're in, you can be infectious at all 5 stages. Doesn't matter whether you feel no symptoms, or you're really sick, or you're recovering. You are infectious, meaning you can still give that pathogen to someone else in all 5 stages.
What happens when this, once you start antibiotics 24 hours later, that you're not going to use?
Because look, here's time right here. During this part here, this is where you start your antibiotic therapy, and you still have bacteria there. They're declining, right? They're declining, but they're still there.
But I'm just saying that the doctors would always tell me with my kids that 24 hours of antibiotics and go back to school.
The bacteria is still there, right? You have killed the less virulent ones, right? The easier ones you killed, those guys with the harder bacteria, they're still around. Those are the ones you really want to get rid of. Okay? So not after 24 hours. You can still pass on that infection even after convalescence, where you have no symptoms at all. Okay. Because the line is not at 0. See that? It's not down there. So you still have microorganisms present, correct? And you can still give it to somebody else at that point.
Does it ever get back to 0?
Yeah, it does. It does. Yeah, it will. It will get back to 0. Just over time. We don't have to spend too much time on here. Here it is basically incubation periods, right, between infection, right, and the first sign of symptoms for different diseases. Check out Staphylococcus foodborne infection, right? Less than a day. So Debbie probably had that the other day. Poor Debbie. She was sick last class. We missed her so much. She had McDonald's and she couldn't make it, okay? It was quick and swift. She probably had Staphylococcus, maybe even E. coli in that hamburger. Who knows? Less than a day between what? Infection and her first sign of being sick in the car. Okay. Influenza about a day, but check out things like, of course we know about HIV, right? Almost 10 years, right?
So you can't hang out with somebody who has leprosy for 30 years because you might get it?
Yeah,
That's horrible.
yeah,
You can have it now, I don't know.
exactly. Or hepatitis, HIV, we know about that, right? People can have it, not have any symptoms, but they can sure enough give it to you, right? Yeah, absolutely, absolutely. Now, this slide basically is talking about what? how can a pathogen be successful? In other words, for a pathogen to be successful, it not only has to multiply in you and make you sick, it has to get out of you, right? And get to another host. Make sense? If they stay in you, they eventually will die, right? So they don't want to die. That's a poor pathogen, right? They got to get out of you to get to some other host so they can keep on multiplying. So how do they get out of you? Through portals of exit. Guess what? Their portal of exit is what? The same as their portal of entry, right? This is the opposite of how they got in the first place. They get out the same way. Now, lots of times, the pathogen can get out through secretions or excrements, right? So let's take a look at that. All right, for example, these are talking about secretions or excrements. We know that pathogens get out from the saliva or sputum, okay? We know they can get out in your blood or for secretions from the vagina. How about your nose? You blow your nose, right? You got a cold. The viral particles are in that discharge there. When you cough and droplets, they're in the droplets there. Even in your tears, they're in your tears. So basically anything that come out of your body, whether it's urine or feces especially, it is potentially contaminated with the pathogen. Tears, blood, mucus from the nose, or sputum from the lungs, it can potentially be what? Contaminated with the pathogen. All of these things are basically saying what? How does the pathogen get out of you? They've got to get out. Or they're a pretty bad pathogen, makes sense? They're pretty bad. Now, in terms of transmission, in other words, so what? So what the pathogen is in your saliva, right? So what the pathogen is in the sputum? So what is in the feces? So what? How do we, I know, Rhea, but it's true, right? So what? How do we actually transmit the pathogen from one host to another? Well, there are 3 types of transmission based on how we're doing it. One is contact transmission. This is where you, the person, you are actually contacting either the host or the reservoir directly that has the pathogen. Make sense? So you're contacting the person, right? Or the reservoir that actually has the pathogen. Now, in terms of contact transmission, it can be direct, indirect, or a droplet form. And of course, I'll explain all those in the next slide. Vehicle transmission, think of your car, right? Your vehicle transports you to school, right? So in vehicle transmission, you're using some agent to actually transport the pathogen from one host or a reservoir to another host. Make sense? So vehicle transmission, you're using some agent, think of a car, to actually move or transport the pathogen from either the host or the reservoir to another host. And lastly, we've got vector transmission. We talked about vectors about 2 chapters ago, right? You've got mechanical vectors and biological ones. That will be a recap, because I taught about that a couple of days ago. All right. Now this table here, 14.11, 1411, it summarizes all of that. And I'll go ahead and jump here. Under vehicle transmission, please write in your notes, basically body fluids, okay? Body fluids is another type of transmission in terms of getting the pathogen from the host or reservoir to another host. So input under vehicle transmission, you got airborne, waterborne, foodborne. Please write in bodily fluids as well. Okay. Let's first talk about contact transmission. You're basically contacting something directly, like with your hands or your mouth, for example. In direct contact, okay, it's direct. So you either shake someone's hand, right? You sneeze on it, you shake their hand, you put your hand to your eyeball. Okay, great, you got a cold. Or you kiss somebody, right? Or you have sexual intercourse with somebody, right? Without using protection. All of these are what? Direct contact. Even bites, for example. Hopefully not from a person. But bites, okay, they are direct contact. So things like genital warts, gonorrhea, herpes, all your favorite STDs, right? Direct contact diseases, okay? Direct contact. Okay. How about indirect contact? In indirect contact, this is where you are touching a fomite. So that's F-O-M-I-T-E. So a fomite is basically your inanimate object that has a potential pathogen on it. So you touch it. Think of things like money, for example, pencils, the table, toothbrushes, cups, anything like that. These are fomites, inanimate objects that can have the pathogen. When you touch those guys, okay, that's indirect because you're not touching the host or the potential reservoir. Okay? So again, don't drink after people, right? Don't use utensils after somebody. Don't use someone else's toothbrush, right? Because it can have a pathogen on there. Toys, money, pencils, etc. How about droplet transmission? Here's the deal. I've got to spell out droplet versus airborne. The key difference here is the distance. So in other words, you cough or you sneeze, right? Those are droplets. Those droplets will contain either viral particles or bacteria in the droplets. Now, if it's a droplet transmission, these droplets are traveling less than 1 meter. So you're pretty close to someone who just sneezed. So that's considered under contact. In other words, I'm right in front of Milton, I sneeze, he's less than 1 meter away from me, I gave him something, okay? So the only difference between what droplet transmission and then airborne is basically the distance traveled. Got it? So under droplet, less than 1 meter. Okay. Now let's go under vehicle transmission. We're using some agent now to transport the pathogen. Okay. Airborne. So again, we have droplets. But these droplets are in the form of an aerosol. Now what's an aerosol?
Spray.
An aerosol is basically the water droplet with a dust particle. That's an aerosol. So an aerosol is the water droplet that you just sneezed or coughed with a dust particle. That's an aerosol, and they can travel more than 1 meter. Typically, the bacteria are actually on the dust particle, or the virus is actually on the dust particle, and that's how they're traveling. For example, where's my favorite one? They don't have Legionella on here. So for example, how do you make aerosols, for example? Okay, when you sweep your house, right? Bringing up dust in the air, right? You form aerosols, okay? Air conditioning units, they form aerosols. I talked about Legionnaire's disease, right? Okay, that's how it got spread, through the AC ducts, making aerosols that had Legionella on them, okay? And it traveled more than 1 meter distance. A lot of people got sick and died. Waterborne transmission. Waterborne is the most important mechanism in terms of gastrointestinal diseases, okay? Lots of them come from what? Contaminated water. Now we see here that water is a mechanism for transmitting diseases, but water is also a what? A reservoir for diseases. So in this case, water is both a transmitter and water is also a reservoir. Think about the fecal-oral route. In other words, if you have contaminated water with fecal material, lots of diseases get into the water just by that way, like cholera, for example. Or schistosomiasis, that's caused by a worm, for example. They get into the water through what? Feces. And then people end up drinking the water. And then they get sick by it. Foodborne diseases, it can come from basically improper food processing. In other words, the canning process didn't use the high enough temperature to kill all the bacteria. Or how about basically poor hygiene, like sushi, or poor hygiene, so the workers in that plant didn't wash their hands. Or poor refrigeration. All of those can be transmitted as what? Foodborne diseases. Let's talk about vector transmission. Real quick, we talk about mechanical versus biological. What's the difference? In a mechanical vector, like your housefly, for example, like your cockroach, they're not going to bite you. Some flies do. But usually what flies do, they hang around feces, right? They hang around dead animals, and they fly onto your pizza. They are carrying any pathogen, right, any bacterium from the feces, from that dead animal, onto your pizza. They just physically carry it with them, right? They land on your pizza, and if you don't shoo that fly away real quick, okay, you eat the pizza with anything they left on your pizza. Yummy. What about cockroaches? Same thing. They like to crawl around icky places, right? They're crawling in your cereal box. You eat your cereal, okay, you're going to get whatever they were carrying, right? Yummy. Now what about biological? It is biological because the actual pathogen completes part of its life cycle in that vector. That is key, okay? So in the biological vector, that pathogen actually completes part of its life cycle in the vector itself, like in mosquitoes. In other words, we talked about, briefly, we mentioned malaria, okay? We understand that the Anopheles mosquito is the vector for malaria. Okay, Plasmodium has most of its life cycle in that mosquito. And then it completes its life cycle in you, okay? So that's the difference between biological, right, versus mechanical. Oh, boy. Now this young, This is what happens when you sneeze, okay? Now, if you're standing right there, oh boy, that's less than 1 meter. That's what droplet transmission. I can see your hand, that's droplet transmission.
That's aerosol.
It will be an aerosol later on, down the line,
if it
right?
was
Down the line.
more.
Down the line. When it combines with dust in the air, like someone's sleeping, okay, that's now your aerosol. But if you're real close like that,
This is spray.
she's spraying. Then when you sneeze, don't sneeze into your hand, right? No. Do that. Or get a little tissue, right? Because if you do this in your hand, you start touching things, right? And you leave it for somebody else. So sneeze right here, right? Because you don't touch things with the inside of your elbow, right? Or if you got tissue, sneeze into your tissue. What's your question, Katie?
What's the difference between typhoid fever and typhus fever?
The
the same?
bacterium.
Okay. Do they cause the same thing?
No.
Okay.
Typhoid fever is caused by salmonella.
Okay.
Okay? Typhus, I forget the actual bacteria that causes it, but it actually uses a vector, uses a tick to transmit it. That's the difference.
Okay.
Typhus is typhus, typhoid is typhoid. Completely different bacteria cause it, and completely different modes of transmission. Salmonella is typically foodborne illness, okay? Typhus, you need a
A
tick
vector.
to
Okay.
bite you.
Okay.
Look it up.